Good afternoon, everyone. Good afternoon. Welcome to the 2025 Don P. Giddens Inaugural Professorial Lecture Series. My name is Ed Schlesinger. I'm the Benjamin T. Rome Dean of the Whiting School of Engineering, and it is truly my pleasure to be here today as we honor Sri Sarma, a professor of biomedical engineering, Vice Dean for Graduate Education at the Whiting School of Engineering, also a member of the Data Science and AI Institute, Associate Director of Hopkins Institute for Computational Medicine. She founded and directs the Neuromedical Control Systems Lab, is the Executive Director of NeuroTech Harbor, and sits on my Executive Council as an advisor. So, part-time work. But she does an awful lot for the school and the university, and it really is an honor to be able to be here today and present her in this initial professorial lecture. Sri's research develops computational, data-driven, and biological approaches to advance the knowledge and treatment of diseases of the nervous system, including epilepsy, chronic pain, Parkinson's disease, and insomnia. She also harnesses dynamical systems and control theory to understand how the brain processes information and governs activities such as motor control and decision-making. Her talk this afternoon will explore how we measure the health of the brain by developing a standardized model to be used as a global tool to evaluate and assess brain health. We can quantify the effects of disease and separate healthy brains from unhealthy ones. That tool is called the Brain Entropy Index, or BEI, and is being developed at the Sarma Lab. And further, her lab developed EpiScalp, which models local brain networks to distinguish something like epilepsy from other seizure-mimicking disorders. Her work utilizes concepts for control theory, physics, and data science to leverage the union between neuroscience and medicine to improve human health. Clearly, she is doing some of the most advanced, cutting-edge research in this field, and it is an honor to have her as a member of our faculty. Before we hear from some of the others and Sri herself, I would like to take a moment to recognize one of my predecessors, Don Giddens, for whom this inaugural professorial lecture series is named. Don Giddens was the 5th Dean of Engineering at Johns Hopkins and a Professor of Mechanical Engineering. And now it's my pleasure to introduce Mike Miller, the Bessie Darling Massey Professor and Director of the Department of Biomedical Engineering here at Johns Hopkins University. Mike?
There are so many great things about being a department director. Sri is one of those. I'm so fortunate. We are so privileged to have Dr. Sarma in our department and our university. Sri and I go way back, and I was thinking about that, and it was so nice to meet Sri's family today because this is the first time that I've met you, and I really appreciate your coming. Yet we've talked about your family and we've talked about many things. And one of the things that really means something to me is, do you remember that time we were walking in New York City? I think it was part of the first Kavli, when the Kavli Prize was given, perhaps, or it was another event. And we were talking about your having come from MIT. And of course, Sri received her PhD around 2005, 2006, I think, is what I remember, at MIT, from a very famous laboratory. And that's how I would have known her. I knew her mentors. There's Ron Daniels. Welcome, Ron. It's great to see you. Thank you for being here. And, I was thinking when we were walking that day, we were talking about what it means to be an academic professor at Johns Hopkins, and how one of your mentors, who I knew well, had said to you, Johns Hopkins was a great place for you. And we both agreed, and it's been a great place for me. Now, I've learned many things from Sri along the way since 2006. Here are two of the things. Every time Sri gives a lecture about the brain, I learn about control systems, and I learn about observability and controllability, which I actually learned from Jack Rue here as a student. But of course, you learned from some of the sources of that theory. You know, MIT was really creating those theories. But the other aspect of what I learn every year is that we teach a class together, a core class that all 100 undergraduates take. I teach the first half and she teaches the second half. And I always feel that the students respond so well to Sri's half of the class. And they always talk about how great Sri is. And of course, what we had to do, instead of teaching co-located, we had to split it up across the Christmas vacation because Sri realized that my ego was really struggling with our students. So we learn from Sri all the time. And as a department chair, one of the other things that I've learned from Sri is how to interact on human levels. So every time we have a department meeting, if Sri can't attend in person, she'll either call me or text me and say, Mike, I'm sorry I can't be there today. I'm going to be in Washington, but I'll be on the Zoom. And I so appreciate that. So thinking back after that walk in New York, I was thinking about how you were recruited. And I believe that you were recruited through the formation of the Institute for Computational Medicine. Now, some of us are aware of this, but Johns Hopkins was the first to have computational medicine. That's now been copied all over the country. And at that time, there were really 4 great centers and institutes that I was aspiring to be part of. ICM was one of them. Of course, Fred Jelinek Center was one of them. Russ Taylor Center was another. And of course, the Robotics Institute was. And so ICM was one of the great centers that was emerging. And so Sri was recruited to be part of ICM and as well biomedical engineering. And I thought it would be nice if the director of ICM, Joel Bader, would come up and talk a little bit about Sri's contributions to the Institute for Computational Medicine.
So thank you, Dean Schlesinger. Thank you, Mike, for giving me the opportunity to speak. And thank you, President Daniels. On behalf of the Institute for Computational Medicine, I want to first welcome Sri's family and friends, and of course, congratulate Sri. We are all so happy to have you and so proud of your accomplishments. Sri is unique in bridging scholarship and entrepreneurship. That's important because in the lab, whether it's a computational lab or a wet lab, we create new scientific advances and engineering advances, but part of the mission of some of us is to bring those advances to society. Crucial for medical advances being made by Sri and other faculty in the Institute for Computational Medicine. Sri was 20 years ahead of her time, and after her PhD, not going on to a postdoc, and instead co-founding a startup in data analytics, which we would now say, AI and machine learning. Somehow, she decided to come back to academics. So our gain is somewhat their loss. In doing a postdoc and then joining the faculty at the Institute for Computational Medicine. Now, though, she has a fantastic role as a leader of NeuroTech Harbor, as a mentor to junior faculty and groups that are trying to come together and bridge the gap between research papers and real-world impact. So I have a clear memory of meeting you for the first time in person at dinner with Ray Winslow in the cafe that is now another restaurant. And you were as energetic and optimistic then, as you continue to be now. And in our institution, from biomedical engineering to the Institute for Computational Medicine to the Whiting School to the Johns Hopkins University, are just so lucky to have your leadership. Thank you so much. And now I'm told I'm supposed to advance the slide.
Good afternoon, everyone, and welcome to Dr. Sarma's inaugural professional lecture at JHU. I apologize for not being able to make it in person because of my travels. It is indeed a pleasure to introduce Dr. Sridevi Sarma for this important lecture. I really would like to commend you for this tradition, giving everyone an opportunity to learn what their colleagues are doing in this particular fashion. I wish that MIT would adopt something like this. It is indeed one of the high points of our careers as faculty and mentors when we get the chance to introduce one of our star students. Many of us joke that the PhD journey is a 5-year process of us cloning our students in our images. Then when they succeed, we are very proud, sort of a success for us. But Sri's trajectory in academia has been quite fascinating and quite unique. As a PhD student, she spent her time thinking about how feedback control systems behave under non-ideal conditions. For example, when signals are encoded in nonlinear ways before they are passed through the controller. That's a problem that emerges, for example, in remote control. Think the space station or the Mars rover, or think about teleoperation where you're conducting a surgery from a distance. During that time, she also minored in neuroscience out of sheer personal interest. I had no idea at that time that neuroscience would become her passion and the field that she would pursue her future academic work. So as a postdoc and a faculty member, Sri transitioned from being a theoretician into becoming a neuroscientist and, in a way, a systems and data scientist, applying her skills to various challenges about the brain, and one who focuses her research on outcomes. Because of that, she tackled big problems using her lens of systems and feedback, ideas about robustness, ideas about feedback control, certification of guarantees, all the basic stuff that she learned as a control theorist, but collaborating with experts in the domains. She defined a new direction for herself that has now become trailblazing for students who come after her. Her work is amazing, and she got many accolades to recognize her work. I would say, just mentioning two, the Presidential Early Career Award for Scientist and Engineer, the PCAS, which is very competitive and prestigious, as well as her 2023 prestigious and competitive NIH Outstanding Investigator Award, just to name a few of her prizes and awards. Needless to say, she has been invited to give keynote lectures in many institutions and conferences around the world. Now, I'm not going to spoil the surprise here on what she's going to talk about, but I do want to say that Sri has emerged as a superb communicator and obviously a teacher and a mentor. If you want evidence, well, stick around and you listen to the lecture today, but also you can tune to her episode on brain games. The title of the episode is Gambling Brains, where she ties the whole question of decision-making to the state of the brain. It's a fascinating episode and actually directly comes out of her research program, but she explains that in a way that anyone could understand how these two things interact. Certainly, because of her excellence in education and mentorship, she was given many awards, and I would mention that there are 2 awards that she received from JHU. One is the School of Engineering Robert B. Pond Excellence in Teaching, and the other one is the Capers and Marion McDonald for Excellence in Mentoring and Advising. Both are testaments for her dedication to teaching and generously giving her time for her students and postdocs. Sri's interest did not stop at the scientific knowledge discovery. She also realized her vision through translation research and producing products. Her company, Neurologic, aims to address the problem of detecting possible future epileptic seizures. Now, what is most interesting is that the product is an outcome of her work viewing the brain as a network and as an interacting network and analyzing robustness or fragility at specific nodes in that particular network. It is amazing to see that piece of theoretical work, seemingly esoteric, turns into a product that can save lives. So you would think that this is all enough, but not for Sri. She has a lot more energy for that. So during the pandemic, and while I say many of us were pondering the existential threat upon us, she was active and she took a big effort to support and launch the NIH NeuroTech Harbor, which focuses on funding, giving a large amounts of funds for translation research. Before that, she also accepted to become the Vice Dean of Engineering responsible for education. It is not just reading her CV. It's just I'm amazed by her ability to handle all of this, and while giving her time to her graduate students and postdoc, raising her family, and still keep her positive and joyful outlook. So I want to close by simply coming clean and declare my failure in cloning Sri in my image. And I do think, and I admit, that the world is a better place because of that. Sri is unique. She's a unique person that has defined herself distinctly from her colleagues. She's a brilliant faculty who has and will continue to make substantial impact across research and education. It is my pleasure and honor to be associated with her long and successful career. Thank you.
So that was my mentor. That was my PhD advisor from MIT. He's a professor in electrical engineering and computer science, and he was the founding director of their Institute for Data Systems and Society. When I was a PhD student, I was a pure electrical engineer all the way from bachelor's all the way to the PhD. Control theory. I'll talk a little bit about what that is in a second. But essentially, I spent years minoring in mathematics, proving theorems, taking robust control, multivariable control, nonlinear control, introduction to linear systems. And even though introduction is in that title, that was the most difficult class I've ever taken in my life. So with that said, I do none of that today, okay? I study the brain. However, I take some really, really important lessons from that field, okay? And one of the important lessons I learned is that when you want to control a complicated or a complex system, you don't actually need to understand its dynamics in order to effectively control it. So think of riding a bicycle, right? You're the controller, the bicycle's the system. I'm pretty confident that none of you are computing the equations of motions, right? This is one of 4 pages that describes the equations of motions of a bicycle, believe it or not, right? So how are you able to maneuver on this bike? Well, it's very simple, feedback. Okay? So you're using vision to see where you're going, and you're using how the bike feels, proprioception, right? And with that feedback, you make very tiny adjustments, right? What do? You move the handlebars, you speed up, you slow down, right? You might move your body. What's the goal? To stay upright, move forward, and maybe follow a path. Okay? That's control. Now, riding a bike is easy because we teach our kids. This is actually my son. I think he's around 3 years old, and my friend asked, is this slow motion? No, this is not slow motion. This clip was done on purpose. There's kids whizzing by, but you don't see that here. And of course, me and my husband who are here are thinking, he's our son. He must be simulating some of these dynamics because he's just our son, right? So all sits aside, yes, he's now graduating from University of Michigan, and he's just cautious. That's who he is. So with that said, the lesson I learned, the guiding principle of everything I do in my lab is feedback and simple models. To what? To understand complex systems and figure out how to control them. And this complex system of interest that I've been interested since after grad school, is the brain. We're very interested in building models, simple models, to understand the brain in health and in disease. When I first came to Hopkins, I was studying Parkinson's disease, and in particular, a treatment called deep brain stimulation. You put electrodes in the brain and you change the activity pattern so that you can reduce their symptoms. Again, the philosophy was simple models so that we could figure out how to do closed-loop deep brain stimulation. And I had plenty of collaborators. More recently, I work with several folks at the School of Medicine in the Pain Institute, and we look at chronic pain. And we're building simple models of your spinal cord and your brain to try to understand how does peripheral nerve stimulation change and alter activity in the pain system. And the goal is to reduce pathological pain in chronic pain patients, right? But today, here's what I'm going to talk about, because I think everybody in the audience can relate. We all have a brain, and we all are potentially more and more concerned about whether we have a healthy brain as we get older. Right? And so today, I'm going to talk about a large body. We do a lot of things in the lab in terms of biomarkers, right? We try to build simple models from imaging data so that we can understand the brain in health and in various disorders. Why? Because we're trying to give better information to the clinician so that they can better diagnose and better treat their patients. Today, I highlight two things. So I'm going to focus on brain health. What's a biomarker to just compute whether your brain is healthy or not? And then I'm going to move more specifically to some of our work on epilepsy. So when you think of biomarkers, right, many of you probably are thinking blood tests, right? You go to the doctor, do a lot of things. They check your vision, they check your hearing, they check your weight, your body mass index, and so forth, and they take your blood, right? And blood is one of the richest sources to come up with disease biomarkers, right? It reflects almost every single organ in your body. Right? So thinking proteins, antigens, things you can compute from blood cells, right? And to see whether do you have a healthy heart, or do you have cancer? Do you have an infectious disease, autoimmune disease, something in the endocrine system? And all these biomarkers satisfy very specific criteria, right? A good biomarker is something that can clearly distinguish between health and disease, right? It should scale with severity, right? You think of these numbers they compute for your cholesterol. The higher it is, the worse you are. So it should scale with severity, and it should change with treatment. This is how we can test whether a particular treatment, be it a drug or stimulation, is working or not. But the question we're asking now or today, and this is actually very fresh work, unpublished, okay, is what about the brain? Can I come up with a biomarker for brain? And we're not going to do blood biomarkers. There is some work under development in terms of trying to come up with a blood biomarker for brain health, but it's not really, it's very much in that infancy. Okay, so with that said, why do we want a biomarker for the brain? Well, the first thing is once you hit adulthood, over 40% of adults worldwide suffer from some neurological disorder or disease. Okay? And actually, they often get diagnosed only after symptoms emerge. So neurology, I would argue, is often a very reactive medicine. Biomarkers are supposed to allow you to be proactive, but in neurology, especially when it comes to brain disorders, we're very reactive. And this is a problem. Now, one of the pie charts I show on the right is sort of the global burden of neurological disorders in percentage of disability-adjusted life years. The big one is stroke. Right? And the next largest is the Alzheimer's and the dementias and so forth. So this is a real global burden. So that's why we're after a biomarker for a healthy brain. Ideally, we want a screening test. Something when you go for your annual exam to your primary care physician, they're not just testing your eyes and your ears and your heart, they're actually going to test your brain. And can you do that? Can you do that in minutes and actually get something reliable? And can it be a nice screening tool that ultimately will detect that you're going to eventually have a neurological problem in adulthood? So that's our goal. The work I'm going to start off talking with is the work of my PhD student, Luis Sanchez, who's here as well. And if you have any complaints about this work, you can talk to Luis. Okay, so what do we do in the doctor's office today? You all have experienced this in terms of measuring health of your nervous system, right? What's the knee jerk? The knee jerk reflex. They hit you with a hammer, and this checks the reflex whether your nerves and your spinal cord are working properly. And some of you might have even undergone this test of coordination and strength, and what they're doing is they're looking to see if one of your arms dips or pivots in. And that could be a sign of poor brain health. And conversation, things like if your mood, you're not feeling well, they'll ask you about that. They'll ask you about your family history and so forth to check for mental health disorders. But it's so interesting because none of these are measuring anything directly from their nervous system or your brain. And I say that's interesting because there are several technologies, non-invasive and invasive, that actually directly measure brain activity. So why aren't we using this technology in the doctor's office? Okay, this is an example of EEG, electroencephalography. You wear just a cap and it's got contacts, so electrodes, just like you would for an EKG, doesn't hurt, and it captures brain waves or voltage activity on each of these electrodes. This is an EEG scan, okay? And our vision here is to take that scan and within minutes compute one number, okay? A brain health index. We're going to call it BEI, comes from brain entropy, and I'll get there. But a number that's between, say, 0 and 1, where if you're above some threshold, your brain is normal, you're okay. But if you're below that threshold, it's a sign of a disorder, and maybe you should go see a neurologist, okay? So that's the goal. So with that said, we're in the era of data science, AI. We have DSAI here at Hopkins, a big initiative, large language models, deep nets, all this stuff. And you might say, okay, one way to approach this problem is I'm going to take a bunch of EEG scans, and there's plenty of data available in the world, publicly available data of brain imaging scans in healthy people and in people with neurological disorders. So we could do thousands of scans, put them into a big machine learning model, and come up with a number. That's not what we're going to do today, okay? Not that it's a bad approach, but I would argue that it doesn't really teach us a lot about what's going wrong. And then the question we're after is, well, what are properties of a healthy brain? What does it mean to have a healthy brain? And I tap into what I learned is we seek a simple model, okay, to answer this question. So what we're going to do, develop this metric I call BEI, and we're going to combine 3 fields: thermodynamics, brain imaging, and statistical modeling. And I'm going to keep it at a reasonable high level so everybody can follow. Thermodynamics. So here's the roadmap of how we're going to develop this measure. The first thing we're going to do is talk about entropy, okay, and how entropy connects to a healthy brain, okay? And with this entropy hypothesis, which I'll go through, we're going to figure out what properties emerge that are characteristic of a healthy brain. So from entropy, we get properties, 4, and I'll talk about them. And then we're going to say, okay, if I know that these are 4 properties that a healthy brain should exhibit, can I measure whether those properties are there from an imaging scan, from a brain scan? We're going to talk about metrics. And then we're going to move on to, okay, if I have ways, 4 numbers, 4 metrics to capture these 4 properties, can I find an optimal combination to give me that one measure of brain health, the BEI? Okay, so that's where we're going to go. So let's start with thermodynamics, okay? I start with the founder here, a physicist, Rudolf Clausius, who was the one who summarized the second law of thermodynamics, that entropy is increasing and it tends to a maximum. And I will define entropy very high level in a second. But I like this quote, which comes from an astrophysicist, Arthur Edkington, and I highlight something in particular, basically saying that, you know, entropy always increases, always hold. And it's basically the supreme position of all laws of nature. All laws of nature should probably follow this principle, right? And he even says, if your theory is found to be against the second law, I can give you no hope. And I think Luis was taking a class where they were talking about entropy, and he was connecting the dots and said, wait a minute, I think this might have something to do with the brain. Okay, so let's talk about entropy, okay, in a very simplistic way. So entropy is a measure of atomic freedom and randomness, okay? And basically what it's doing, it's measuring how atoms in a substance are free to spread out, okay? Spread out, move around, arrange themselves in various configurations or in unpredictable ways. Okay, so you can see here, as you spread out, entropy increases, and that's why this is a simple example that when you go from a solid, where molecules are very ordered, okay, and organized, to spreading out and becoming more random, entropy increases. Okay, so that's the basic concept in thermodynamics. And we posit that actually the brain also may have this principle, adhere to this principle. So what do we mean by that? So we have the brain at work and the brain at rest. What do I mean by the brain at work? That's when you're solving a math problem, reading, moving, and so forth. And what's going on in your brain then? The neurons and the neural activation and the neural energy is more ordered. It's localized. It's specific networks that govern those behaviors, right? But then the question is, when you're at rest, when you're not engaged in any task, and I know that's an ideal setting, you're not engaged in any task, then the neurons, we believe, their activation should spread across the entire brain, okay? Almost like background chatter. They're not zero, flatline, because then you're dead, but they're still alive, but it's just background chatter completely spread across. Why? Because this is a sort of an efficiency argument here, and I'll get to that in a second. So that's the idea, and here's the hypothesis that we test, explicitly stated. So our argument is that if you have a healthy brain, okay, then at rest, all the energy should be spread about. It should maximize entropy, okay? And why is this? Because this kind of reflects a state of functional efficiency, right? So this enables rapid reconfiguration of activation in your brain in specific networks when you need to immediately do a task. Think of a predator-prey scenario. The prey is grazing on a grass at rest, right? And the minute a predator comes, that animal has to pounce, okay? So the question is, how do you immediately activate the motor networks, the sensory motor system to pounce so that you save your life? And the argument is that the best configuration at rest is when everything is spread out, okay? So this is not actually new, and maybe it's not surprising to many of you because lots of biological systems and organism systems have this property of when they're at work and they're coordinating and they're talking to each other, entropy is low, they're coordinated. And then when they're freely roaming, their entropy is high and they're not talking to each other necessarily. Think of fish just, swimming around. What happens? When there's a predator, they organize, right? And this organization is a drop in the entropy, okay? But when there is no predator, kind of at rest, they're freely roaming. They're decoupled, if you will. Similar to birds and swarms of birds. This is slime mold. I never heard of it till I was searching around. Slime molds, they exist in moist environments, like in logs under the forest, and they're individual cells, and their job is to find nutrients to stay alive, right? And when they are at rest, they're actually all looking for food, okay? And they're not really talking to each other or communicating. But the minute one of these cells finds a nutrient, they actually couple and they form a network. And then they spread out the nutrients throughout the network, so they feed each other, okay? So going from low entropy to high entropy. And finally, your immune system. Think of T cells. When you're perfectly healthy, no injury, all these cells are spread throughout the body. But the minute you get an injury, what happens? They converge to the injured site and they go to work. Again, a state of low entropy to high entropy, okay? So now we're gonna make this more formal. I wanna maximize entropy. The claim was the distribution of energy across the brain should maximize entropy. So let's take that literally and formulate the problem. So we're going to focus on Gibbs entropy, and this is only one of two equations I have in my talk. So this is the first one. So what is S? S is entropy, and it's this integral of a distribution P of E. So E stands for energy. It's a vector. Why? Because it's energy of region 1, energy of region 2, and so forth. So you have a vector, and P is a probability density function of that. Oh, there should be an ln here. This is log. So this is your entropy. And what we're trying to do is we're trying to understand what distribution P will maximize entropy, okay? This particular cost function. What does P, which distribution gives a maximum value here? But we have constraints that we have to, when we solve this maximization, right? The first is that P should be a legitimate probability density function, which means it integrates to 1, okay? The second one is energy. Energy is non-negative. You can't have negative energy, okay, unless you're thinking directionality. But you don't have negative energy, so it's non-negative. And then we constrain the mean, vector, and the covariance matrix of the distribution to be some value mu and sigma. Now, this optimization problem was solved decades ago. This is not a new problem, okay? But it was a formulation based on this hypothesis that we're going to test, and it's been solved. And what's the solution? Well, three things emerge as properties in this solution. The first says that the distributions are IID, independent, identically distributed log normal. So let's talk about log normal. This is the shape. If I had just one brain region, this is the distribution of energy. It follows a log normal, this is the mathematical expression of a log normal density. This is a two-dimensional version of log normal. So that's one thing. Independent, what that means is that the energy across all these brain regions are independent, statistically independent from each other, meaning they're not talking to each other. There's no coupling across the brain. Again, in an ideal setting at rest. That's what the theory says, okay? Identically distributed means all the distributions across my brain regions should look the same mean, the same variance, all log normal, okay? So that's okay. So once we did that, we're like, okay, log normal. Interestingly, we found a paper 10 years ago where neuroscientists actually recorded neural activation at multiple scales in the brain. And what did they find? That when they just plot what they observe in terms of neural energy, it's log normal. When you're even if at the tiny microscale of when two neurons connect through synapses, log normal. Neurons, multiple neurons, log normal. Populations, thousands, hundreds of thousands of neurons, log normal. So this is just observations, but our theory actually says if you're healthy at rest, you should have a log normal distribution. Another observation that's important is what goes on in disease. So I mentioned that we studied Parkinson's disease. Well, one of the properties of a healthy brain is if you're not engaged in a task, the brain regions shouldn't be talking to each other. Their activities should be completely decoupled, right? And this breaks in disease. So think of Parkinson's disease. So this is one of the primary symptoms of Parkinson's is motor symptoms, and one of the hallmarks is tremor at rest. They call it tremor at rest. Somebody's sitting, they have no intention of moving, and one or both of their arms or legs has a 4 to 6 hertz oscillation rhythm, tremor. That tremor comes from specific hypersynchronization of neurons in specific parts of the brain. So here's a disease where one of the phenomena we see is that at rest, there's coupling. So it breaks the property of health there. And of course, seizure. If you do nothing, some patients can have seizures, and that's hypersynchronous, very coupled brain regions, okay? Again, coupling, disease, associated with disease. So to summarize this, here's our conclusion, that if we take the hypothesis that the brain at rest maximizes entropy in terms of energy across the brain, it has these 4 properties. The distribution should be log normal anywhere in the brain, if you look at energy. They should all have the same distribution, the same mean, they should all look the same, okay? They should be independent, meaning there's no coupling or correlations between energy here versus energy there, right? And then finally, this is the last piece which I didn't talk about, which is efficiency, which I mentioned that there shouldn't be some kind of efficiency. And in this case, there is some prior art where at the end of the day, if you're thinking of a healthy brain, at rest, first of all, the energy shouldn't be high. If you're producing so much energy at rest, that's not efficient. So that shouldn't be the case in health, right? And you should also have very little fluctuations of energy and energy. So your variance should be low. And so having the mean and variance low is an efficiency that we believe happens in health. 4 properties, okay? Now we're gonna go to part 2. So if these 4 properties are true, let's first then say that's great, if it's real, right? But then how do I measure this from brain imaging data? So let's talk about that. So the first thing is energy. Usually neuroscientists don't speak the language of energy. They talk about neural activation, firing rates, action potentials. So let's talk about energy, because that's where our theory is, okay? So if you talk about how do I measure energy of one neuron? A neuron is an electrically and chemically excitable cell. If you see a flash of light and you have a neuron in your visual system, it will fire an action potential, means the voltage across that membrane shoots up, okay, and causes a spike that you can measure with a microwire, okay? Somebody computed that the ATP cost per action potential per spike, or the energy, is 8 times 10 to the minus 14. That's for one neuron. And then of course, if you think of energy of tens to thousands neurons, it's very simple. You just compute the number of spikes you get across all these neurons and multiply it by 8 times 10 to the 14, okay? and that you can compute off of microelectrode arrays. Now, if you measure more population level, hundreds of thousands, like you get with EEG, okay? What do you do there? Well, that's where we actually say, look at energy being the integral of power, voltage times current, okay? Or use Ohm's law and you have the integral of voltage squared over the resistance. Just talking about how we compute energy from what we can measure in the brain. V is the voltage. We can measure that with EEG. Finally, functional magnetic resonance imaging, fMRI. This measures blood oxygen levels. It's a correlate of the voltage. That's why I have the bold being a correlate of ET. But in a similar fashion, here is how we can compute energy from a brain scan, a time series, like an EEG. We simply say that the energy in some region I is the integral or the summation of the voltage squares. Where did the resistance go? Well, we normalize it. We divide by the maximum energy and assume resistance is all the same. I know it's an assumption. And we lose that variable. And so we just do energy being sum of squares. Here's how it looks like in sort of a visual form. So if this is an EEG, I have all these circles are electrodes. Each of them give you a signal like this. And what we do to compute energy is we look at a single window W, and we basically take the whole scan and partition it into little windows of size W. And then what we do is in each of those windows and in each channel, we compute energy as the voltage squared, right? So just take that signal, which is voltage, square it and sum it up. And that gives me the energy in this channel for this window. And we can basically, for each window, compute sample energy vectors, okay, from the brain imaging. Then what we do with that data is we apply maximum likelihood estimation and try to see how well does it fit an n-dimensional log normal. Okay, remember the theory says it should be log normal, so we're going to see how well those samples fit the log normal distribution using maximum likelihood. What you get from the estimate is a mean vector and a covariance matrix. So one brain scan gives me a mean vector and a covariance matrix on the energy distribution, okay? Now let's go back to our properties, and the claim is from that distribution, I should be able to compute all four properties. Okay, so let's talk about that. Okay, so I'm going to do it in pictures. So remember the log normal? Every distribution across the brain region should look log normal. We're essentially computing a metric that is high when you look like a log normal and not so high when you don't have a log normal shape. Okay, so that's property one. Property two, all the energy distributions across the brain should all have the same shape, the same mean, the same variance. So this is this follows the property, this does not. Each curve is a energy distribution for a region of the brain. Okay? Third property, that the energy random variables are independent, they're statistically independent. Well, I can get that from this covariance matrix, because the covariance, the off diagonals of this matrix tell me how the coupling between every pair of regions. So if it's 0 or blue, then I see decoupling. But if it's highlighted and non-zero, I don't have independence. So I can compute a metric to capture what's happening on the off diagonals. Finally, efficiency. Remember, I need my energy to be low mean. I don't want fluctuations, low variance, right? I can get that from the mean vector, which I'm not showing here, and the diagonals. These are the variances. This is low energy variance, and this is higher energy variances, okay? So we have numbers that we can compute from this distribution. So I have 4 properties, 4 metrics. Now what we do, the last part, we take those 4 quantities and we come up with an optimal combination to compute the BEI. What is the BEI? It's defined as the probability of having a healthy brain. And we basically do a very simple logistic regression. The log of the odds is linear in the coefficients that multiply by each property metric, whether you are log normal, whether you are equal energy, whether you're coupled, and whether you're efficient. And the whole idea of maximum likelihood estimation is, what are those weights? What is the optimal combination of weights so that I ultimately get a model, a formula that separates health from disease? The BEI is high in health, low in disease. Okay? Enough of the math and theory. So let's look at some data. Okay, so to basically develop that model, so to train it and test it, held out test set, We're still in progress, but we've so far collected brain scans, imaging data from 525 subjects, healthy as well as patients who have 9 different neurological disorders. We'll talk about that. And then 3 different imaging modalities. One of the things I didn't mention is this metric, we believe, is modality agnostic. Meaning you can get an EEG, or you can get an fMRI, or you can get a magnetic encephalography scan. It should be agnostic. I should still see some proxy of energy equally distributed, maximizing entropy. Okay, so let's look at some example patients, which I think are interesting. So this is an EEG from a healthy subject, okay? And by the way, these are all adults, okay? I have a healthy subject, and this is what the distribution gave me when we estimated the density from their data. We see some obvious things, right? So all the energy distributions, every color is a different brain region, lie right on top of each other. They're identically distributed. If you look at the covariance matrix, it's all blue. They're completely independent. If you look at the diagonal, it's also low variance. This seems to exhibit the properties that the theory tells us, right? This is somebody who has major depression disorder. So we have an EEG scan from somebody who was clinically diagnosed with depression. And what breaks down? A lot of the properties break down. The first thing you can notice is that the energy distributions look different across the brain. They don't all even look log normal. Some of them look bell-shaped. Okay, so you may lose log normality here. Look at the covariance. There are brain regions that are coupled in their energy. I lose the decoupling. And then when you look at the diagonal, there's high fluctuations in energy in this patient. So right away, you can see just from the distributions, things don't look healthy. Here's schizophrenia. I have my schizophrenia colleague here. So here's a patient where, interesting. Okay, so if you look at the covariance, a lot of the regions are decoupled. You see blue, so they're not all coupled. There's a little coupling here, some of the variants. But look at the distributions. They're definitely not log normal, and they're definitely not identically distributed. So different properties break down for this disease. And these are examples from some of the other diseases we looked at. I mentioned we had 9 in this data set. We're looking at more disorders, but we have OCD here, Parkinson's, epilepsy, frontal temporal dementia, and Alzheimer's, okay? Okay, but you might say, Sri, you're cherry picking examples, right? Let's look at the population, okay? So this is a training data, so around 500 patients here. And let's look at each property first. Log normal, how log normal are you across these different 9 diseases and healthy controls? Well, look at that. Healthy controls are not the ones that are most log normal. Okay, let's see what happens. They're still on the higher end, but guess what? The neurodegenerative diseases, Alzheimer's and frontal temporal, have a better log normal shape. Okay, that's one property. The next property, are all those distributions identical? Well, healthy controls, on average, have energy distributions that are more identical than any of the other disorders. And notice here, Alzheimer's and frontal dementia have the least identical distributions. This is the third property, independence. The brain regions shouldn't be coupled. Okay, so this is a coupling ratio, so the lower, the better, right? You can see healthy controls have the lowest coupling. Last, efficiency. This is a metric to capture whether the mean and the variance are low. So when your mean and your variances are low, this measure is high. I have a higher efficiency in health than any of the diseases. Now, put it all together to get that one BEI and test it on a held out test set. Here we go. So this is your BEI. These are healthy controls, and these are the various 9 disorders. Now, interestingly, if you look at the ordering of the disorders, as you go from higher, median to lower BEI, these last 3, they have something in common. Parkinson's, Alzheimer's, frontal temporal dementia. They're all neurodegenerative, okay? If you look at these guys, ADHD, OCD, MDD, what are those? Neuropsychiatric, right? And so forth. So there seems to be, maybe there's an interesting grouping that the range of your BEI might be a sign of what kind of disorder you have, okay? But again, a lot of this is community. Remember I talked about what good markers, criteria that good markers should have? So one of them was it should separate health from disease. The other property is that this should scale with severity, right? So in some of our data sets, we had severity scores, and when you go from healthy to the more severe disease, in OCD, MDD, autism, ADHD, it scales with severity. Finally, what was the last criteria? That this biomarker should modulate with treatment. So if you have an effective treatment, it should go up, right? So we're still working on this, but I have 4 epilepsy patients here. Each of these is an epilepsy patient. This is their BEI. This is the time where we captured their EEG when they were on a medication that was ineffective, not controlling their seizures. All of their BEIs were low. But then over time, all of them were eventually, the doctors figured out what are effective medications, meaning they were seizure-free for 6 months continuous. And look what happens. Their BEI just jumps into the healthy range. So it looks like this BEI is modulating with treatment. So to summarize this, we've shown basically that this metric has promise to separate healthy brains from disease, scales with severity. It may detect efficacy of treatment, but there's a lot more to be done. Remember I said the holy grail is screening. I want you to go to the doctor's office, and if you're 20 years old and you're eventually going to have Alzheimer's, I want to detect that when you're 20. I don't want to have to wait till you have symptoms. In order to test this, you need to do longitudinal studies, and that's on our next steps. Now, one of the things I want to say here is that Luis, the student who's working on this, is building a toolbox. We want to send this to the community. We want them to try BEI on whatever disease they want to try it on. We want them to see if it modulates with whatever treatment they have. We want to put it out there to see if this holds. But the last thing I want to say is, notice I said nothing about if you have a healthy brain, it's this part that has to have this property, this part has... No, we talked about a global set of properties, what's happening globally in your brain, and that should distinguish health from disease. But we've done a lot of work in our lab to find disease-specific biomarkers. And I'll mention just a few minutes to talk about that. For disease-specific biomarkers, our experience has said it's local properties that distinguish between disorders. Not global, but local, okay? So I'm gonna give you an example of epilepsy. So these are 2 children. They're both having a seizure, okay? One of them actually has epilepsy, and one of them does not. Okay, so if you are a parent and this is your child and you're looking at this, the question is, which one of those kids have epilepsy? Okay, well, to really confirm what's going on, you need to capture what their brain is doing during the attack. And it turns out if you have epilepsy, so the right one was actually epileptic, you actually see signatures of seizure, the neural source, which you don't see in this, which is called psychogenic non-epileptic attacks. PNEA. Medications don't work for this type of patient. Cognitive behavioral therapy does, okay? But when you go to the doctor, they might think you have epilepsy because that's what your family witnessed, right? Turns out that misdiagnosing epilepsy is very prevalent. In the United States, every year 1 million people go into the ER claiming to have had their first seizure ever in their life, okay? When they're finally sent to the neurologist on that first visit, they're misdiagnosed 30% of the time, okay? And this is a huge burden because if you tell somebody they have epilepsy and they don't, you're sending them home with a diagnosis, a stigma, and medications that have side effects that are not treating their underlying problem. And in the other case, the other error is you tell somebody that they don't have epilepsy, and they do, you're sending home a patient whose seizures could become more frequent and more severe. So why is it so hard? Because there's no reliable biomarker, okay? What happens when you go to get assessed by the neurologist? They do a physical, they look at your medical history, they ask your family, what happened? Tell me what happened, what was this event like? They do an MRI scan to look for structure, lesions in the brain that could have caused some type of event like a seizure. And then they do the routine 20-minute resting state EEG. They just say, sit still, I'm gonna capture your EEG, okay? Note that they are not having a seizure in the doctor's office at the time. Now, what are the neurologists looking for on the EEG? They're hoping that certain indicators of epilepsy are there, abnormalities that are specific to epilepsy. And they call these spikes and discharges, for example, is one indicator. The problem with these spikes, first of all, they're random. You are capturing 20, 30 minutes from a patient. It's not clear that those random events are going to occur. The EEG might look completely normal and you can still have epilepsy, right? And it's not it's not well defined what this spike is. Doctors can disagree as to whether this is a spike or not, okay? Or artifacts. The minute you blink your eyes, the EEG, frontal contacts go crazy, right? And sometimes they mask the actual abnormality. And also half the time, the doctors don't see anything in the EEG. And so all of this uncertainty leads to this 30% misdiagnosis. So what we did, and I'm almost done, what we did in the lab is we think we came up with an EEG marker that's more reliably diagnosed as epilepsy. And importantly, it does not rely on the presence of any abnormalities. So we're not looking for what the doctors are looking for. Okay, so what is it doing? Well, we think we found a network-based biomarker that's very specific to epilepsy. Let's think about this, just conceptually. If you have epilepsy, focal epilepsy, you have a part of your brain whose job it is to cause seizures, excitation in the brain that spreads, right? So if you have epilepsy and you're not seizing, why are you not seizing? And the hypothesis is that there must be other brain regions inhibiting, holding back that seizure. We call that the local inhibition hypothesis. And that phenomena of some nodes in your brain network being held back and another group of nodes doing the influencing and holding it down look very different from the rest of the brain. And so what we did is figured out a way to identify and detect that phenomena using simple models. Okay, what is simple? This is simple. Okay, it may not look simple, it is simple. It's a system of first-order differential equations. We're basically going to say EEG can be modeled by a simple set of differential equations. I say simple because they're first order. We only have one derivative here. I also say simple because the future, the derivative, is a linear combination of the past. It's as simple as you can get, okay, in terms of ODEs. However, the claim is that this A matrix can exactly tell me, if I look at it in the right way, it'll tell me if I have that phenomena of some nodes being inhibited and others doing the influence. I can actually just look at it and compute a metric from it. We did that. So to test this, we call it EpiScalp. So this is a tool that will take a scan and produce a number from 0 to 1. 1 being highly likely to have epilepsy, 0 being not likely to have epilepsy. And here's a retrospective study. And this was work done by Patrick. I don't know if I mentioned him. He was on the other slide. Okay, so we had 198 patients. Every dot is a patient. All of them were diagnosed with epilepsy on their first visit. Okay? All of them had normal EEG, so the doctors couldn't get anything out of the EEG. And all of them were sent home with anti-seizure medications and a diagnosis of epilepsy. Turns out that later on, upon confirmation of looking at EEG during seizures being continuously monitored, 107 of them actually did not have epilepsy. They had PNEA, the example I showed you, okay, PNEA. And so this was a 54% misdiagnosis in this population. These patients were treated across these centers here. Okay, so what did we do? We ran their first scans, their EEG scans, through EpiScalp to compute a risk score. It should be 0 and 1 here, apologies. And now you can start seeing the separation that the epilepsy patients actually have a higher risk score and the PNEA patients have a lower risk score. And one of the clinicians at Jefferson Hospital told me, Sri, here's how I would use your tool. He goes, because he thinks his accuracy is 98, 99%, okay? So he goes, this, and I'm sure it is, I'm not saying he's not. So he said, here's how I use your tool. If your risk is above this threshold, I'm gonna go with epilepsy, okay? If your risk score is below this, importantly, I'm going to consider not giving them medications, okay? But if your risk score is in the middle, I'm gonna do what I normally do. I'm gonna make the call. And if you use that decision strategy, it turns out what was a 54% misdiagnosis goes down to 17%. So it's not perfect, but it's a huge reduction and could be really important in people's lives when you're sending people home with medications that are not working for them, okay? So this tool, we think it has a lot of advantages of getting the right diagnosis sooner, which means you get the right treatment sooner, okay? We're actually running into, this is now, we have money, so I have a startup company, and we raised money to do prospective trials. So this is being tested at 3 centers. One is Johns Hopkins. We have a PI of that trial here today, Dr. Khalil Husari, who's my collaborator. It's being tested at Jefferson Hospital in Philadelphia, as well as the University of Maryland Medical Center. So they're all, every patient that walks through their door, we're computing risk scores, and then we're going to see how well we do in another year. Now, I just wanted to acknowledge the people who did the work. So Louise and June have been involved, epileptologists, in the BEI work. Patrick, Khalil, and Jorge were part of EpiScalp, okay? I just want to shout out to family and friends, my good friends, and all of these family members are here, except my sister and my kids. They're all in their universities. The dean's office, 5 years ago, I joined the dean's office. I had no idea what to expect. Ed just, I don't know why he picked me, but he said, hey, do you want this job? And I said, what is this job? You know, what do I do? And so forth, and I have no regrets. I learned a lot from Ed, and these are all my colleagues in the dean's office that I interact, Christine and Dan, I interact with daily, and Tina, who does my horrible scheduling. So kudos to her. NeuroTech Harbor, Joel mentioned that, and these are all the PIs and my colleagues at NeuroTech Harbor. And what is NeuroTech Harbor? We're a virtual accelerator. We go out there and we find breakthroughs in neurotechnology, and we fund them, not with our own money, but with NIH's money. Okay, and so we need to know what are the best ideas out there, and we fund them. We've now funded over 45 projects and over $30 million into this, from this center. ICM, Joel and others, this is just some of the core faculty that I've had the pleasure to interact with. It's a wonderful home. We do retreats together, we have lab parties and so forth. It's just a pleasure. And Phelan was somebody who joined on the same week that I did in 2009 here at Hopkins. BME, Mike Miller, right, hired me. I couldn't put all the pictures. I think we have 60 plus faculty or something, but I'll tell you, it's such an honor to be here in BME. These people, when I look around, I don't get nervous with talks, but today was weird, right? Like all my colleagues, President Daniels, Dean, I mean, like all these people are here, and like I have to, like, I don't want to, you know, what am I going to do? Can't make a bad impression. So with that said, and BME, the students, I see some students here, phenomenal students. These are such talented students that are hungry for research. So it's such an honor to be here and take them in. Tech Ventures, I mentioned I had a startup, and I see Christy here leading Tech Ventures, who transformed Tech Ventures at Hopkins when she came over 10 years ago. And these are folks that I work directly with. Linda's here in the audience. Apologies, Linda, she does a lot of the work with the IPs. These are the people who make my job easier to translate. I have a desire to have an impact on patients. I'm not interested in solving theorems or proving that. I want to help patients. And Tech Ventures has told me, you know, figured out how to file patents for me with me not doing so much, just tell them what I did and they take care of the rest, giving me consultants and how do I do regulatory clearance with the FDA and so on and so on. Huge support for me. All my collaborators, Johns Hopkins, we keep saying, oh, you know, it's great collaborations. It's really true. These are all my collaborators, and I think all of them might be in the School of Medicine. Amazing. And then I just want to end with my mentors. These are the 5 mentors in my life. So this is my PhD mentor, which we didn't hear from, but I'll listen. You know, he never, ever gives you positive feedback. So I was like waiting for that video for him to say something positive. And of course, it's never going to happen. It's just never going to happen. But of course, he supports me in other ways in a very strong way. So students, even if your advisor is not patting you on the back and so forth, think of other ways that they support you, because we do try our best to support you in other ways. Emery Brown was my postdoc supervisor. He's the one that inspired me, got me into neuroscience. Ed, you know, the dean's office. I mean, one of the things I have to say with this job in the dean's office is I got to learn not just how the leadership and the council work, which is a highly, highly functioning unit, okay, but I just learned about the school. What are all the departments? Who are all the directors? What are the faculty doing? What are the research? What's going on? And I have to tell you, through this job, I love the Whiting School. I was such a Cornellian. I was undergrad at Cornell, and I couldn't get over Cornell when I went to grad school. And I go to MIT, and I was like, Cornell, and then I go to MIT, and then MIT, I come to MIT, like I can't get MIT, you know. But now it's like, you know, I'm thankful for my education and my training at both Cornell and MIT, but I love Hopkins. Okay, I love Hopkins. And I'll tell you, I still talk to my PhD advisor pretty regularly, and we're kind of competitive at MIT Hopkins. And, many years ago, he talked about the College of Computing, $300,000 donation, and MIT kicks in another $300,000, $600,000 investment. They're going to have a building, a new College of Computing, all this, yeah, great stuff. And then, beautifully, like a week later, Bloomberg, $1.8 million for our undergrads, for financial aid, and any specific undergrads. They're not paying tuition if they can't afford it. That's amazing. So I called him. I said, would you say 600,000? I was like, how about 1.8 billion? And he's like, what are you talking about? So anyways, I love Hopkins. With that said, Ray was the director of ICM who recruited me. You've heard from Mike, and I just want to say, all these mentors have a few things in common, a few characteristics. Brilliance in their fields, amazing leaders in what they do. They're patient. They're very patient people. They're humble. And they taught me a lot. They taught me a lot about the school, about my job, about how to be a good leader, mentor, and so forth, and I'm in debt to them. And of course, our legacy as faculty is not discoveries. We mentor way more students, and the impact is about the students and their legacy and getting them out into the workforce to do good in this world. And so these are all my students over the years, and the ones that are highlighted in green are current. And I had to put this in because today they surprised me for this celebration with the videos and cake and all kinds of stuff. And a student actually these are all my students and mentees across from undergrads to postdocs into a periodic table. So thank you. Thank you so much for coming. I appreciate it. Thank you.
I think you can see why we're so blessed to have Sri as part of the faculty. Amazing researcher, amazing teacher, amazing colleague. Really, it's people like Sri, specifically Sri, that make the Whiting School what it is. So thank you very much and congratulations. And you have to have a souvenir that will, go up on the wall as part of this. So congratulations.
Thank you.
Thank you very much. Congratulations. And please join us for refreshments. Thank you.
Thank you.